This video is on basic epidemiology and virology related to COVID-19. This basic means that I try to simplify for anybody to understand, somebody who has a small knowledge on the viruses and some community public health. So this is the virus COVID-19 I am going to show you here. So it's going through our nasal nostrils into the lung, and here I am going to start with some about the virus and some small knowledge on virus. So the virus is the corona. Corona means that it is its appearance is like a rounded, and it has spikes like this, projections. It's like this, and that's the most important part in the virus, to attach to our respiratory tract. And for the virus, the most important part is this: its genome here. It is coronavirus has mRNA, and this mRNA is a huge mRNA. Unlike other coronaviruses and many viruses, they are kb. The density of virus about 10, 20. Most of the virus is less than 20, but in this particular coronavirus COVID-19 has 30 kb density, and it's a huge mRNA. And it has a capsule that is called, we don't call it as capsule because it's not a eukaryotic cell like a human cell. It has capsid that is consist of lipids and proteins, and this is not formed by the virus itself. It is taken from the cell which is invaded. So in this case it's from human cells. It's from this one. So what the virus is trying to do is, this spike is going to attach to our respiratory lung epithelium, and then it want to give this genome into that cell, and that in the cell we have normal functions, and it is going to invade our normal functions, stop that normal functions, and ask to do whatever they want. What they ask to produce proteins, ask to produce what the way they want. That's what happening with the virus. It's invading, just like enter into a virus attack to a computer. It is going to do whatever the way it wants, not the way we want, not the way we programmed. Little bit similar to that. So in this side, this spike protein, it's with color code showing. So it has two portions. One is a fixed portion and a variable portion. This wider one area is variable portion. That is the one which is attaching to our receptors in the lung epithelium, which is ACE2 receptor. I will tell again. And there's a fixed portion, and in between there's a bond. So it has to attach, and then it has to expose this fixed portion to release this genetic material into our cell and do whatever it wants. So So the scientists, virologists, are doing a lot of experiments on how to attack at this level, the spike, either at the variable, this spike, this area, or fixed area, or and this cleavaging. Remove the, it's like it's to cleavage two parts to expose the fixed portion to attach to our membrane. And this is that. You may remember this, and then we will go into this more details. So here, this is what I was telling. So these are the spikes, and the protein has genome, and then it has to attach to the lung. The lung, it's not like other coronaviruses. I mean the common cold virus. About 15% of common cold virus is corona, and they attach to our upper respiratory tract epithelium. But in this virus, the spikes protein can attach to ACE2, there is angiotensin-converting enzyme 2 receptors in the lung. So the respiratory epithelium has more of this, and the lower respiratory tract has much more. In the alveoli, we have a lot of this one than the upper respiratory tract epithelium. This AC, angiotensin-converting enzyme 2 receptors. So the spike, if it is attached, it's like a key. Spike is going to be key, and the receptor is going to be the lock. It can unlock the two. It's like opening the door, but that alone is not enough. Then there's an adjacent small protein like this on our epithelium in the alveoli, and that's going to cleavage. That's going to break. That's what I showed you earlier. And then it's going to cleavage these two parts of the spike, and then genetic materials enter. So this is the once it enter, this is shown in the you saw that genome. I told you that coil-like thing. It's mRNA chain, and it's coiled up. And this is that the virus proteins. Virus is has only mRNA, and when the other particles of the virus, the ribosomes of our body, our cells, our human cells, attach to that mRNA, and it's going through this chain, mRNA chain of the virus, and producing proteins. So initially it's producing a long protein, and then later this protein is divided into parts and form various functional proteins. So a whole lot of protein cannot do much, so it is going to divide into these colors proteins. For you to understand, this is colors. So various proteins are there. Early it was a big chain, joined one. Now it is going to separate out. These are studied. The various proteins are studied. So the virus itself cannot produce proteins. They get our body cells, and then they produce proteins. So this is that colors. This is that bigger protein chain, non-structured ones, structured ones, and various functional proteins. All are combine. So these are going to separate out, these colors separate out and do various functions. So that's how virus is going to work. Virus is going to attack us. So in addition, when virus is working and doing protein synthesis or proteins, it wants to escape from our body mechanisms. That is, our body can attack to this premature stage of the virus. When it's premature, it is easy to attack, but virus is going to have a what it is like a fort. It's going to have a capsule, double membrane around those things, and then it's like here, and protect the early stage of the virus. Still, this virus, when it is there, when these functions are there, this cell has changed its identity, just like a flag. Say it is a blue color flag, it's normal, and now when it is changed, this flag is going to be blue and red, so that body is going to identify what is this different flag here. It must be a foreign and it's going to attack. Our innate immune system is going to attack. Still, virus also know that, so it has a system here. So the virus produce some proteins inside here. In the case of attack, it's going to delay that attack and delay the innate immune response of the body. That's specific with the COVID-19. So here I told this the virus genome, and it has these proteins. These are the proteins, how it is named, and the outer membrane has this protein, but it has some proteins. These white color ones are not there, so they are inside. So these proteins, this one that is at the beginning top here, NSP1 protein, and that is the one which is suppressing our gene expression and produce its own proteins. Our gene expression is suppressed, and it's going to express its own gene, own mRNA genomes here. And then this is what is happening, and once I told you at the beginning, if I summarize, so the ACE2 receptor is the receptor that virus spike attach. That's without ACE2 receptor it cannot attach. Spike cannot attach. Once it is attached, then there's another cleavage, and it remove the upper superficial part of the spike, and the other part, the inside part, can attach to this membrane and enter into the cell. Then it releases its mRNA here. That's what I wrote. When the mRNA here, then I told you that there's a thing called ribosome here, that is blue color gold, the circle one, speared one, and that's the one which is going through this and reading this virus genome mRNA and start to produce proteins. So when it is like this, then this stage is going to circle all those processes by this double membrane that is really endoplasmic reticulum, double membrane. It's covering, protecting, and once all the proteins are formed, then they incorporate into another virus, and then the way it is entered, the way that it is going out. And one virus, once enter, it can produce 3 viruses, it is estimated. But understand, we have the cells. It's a huge number of cells in the alveoli, huge number of cells. So it's in meters, square meters, several square meters. So the cells number is very huge in the alveoli. So a lot of viruses can attach. So the same process can happen in a lot of cells, and although one cell can produce 3, is much more than that when we get the collective action. So here is the what I told you that earlier, that when the virus is there, that the cell is going to identify. There's a different flag now. The cell is different flag. The express it's expressed in earlier red, for example. Now it is blue and red. So it's different flag. So it's going to produce interferons against viruses to kill the interferon. But this interferon production is blocked by some of those proteins. Some of those proteins which is produced from our own cell, within our own cell, but by the command of the virus genome, it's produced less. And also that's what is showing here in density. It's showing here SARS-CoV-2. It says it's a lower number of interferon, less concentration, maybe less amount, and further, more than less amount, it's delayed. It's going to delay to produce this one. It's not producing at the required moment to attack this virus. It's producing later stage of the virus. See here, when the virus has multiply and it's entered to peak, when it's almost towards the peak here, at this time, the only interferon, this is not clear, but it is saying interferon. When I enlarge it, the letters became faded. So this interferon, the interferon response for other viruses is very early, so it can kill the virus at the beginning. But in this case, this COVID-19 has a protein which can delay this response and reducing its response. And so once it is released, it is not that useful. Only thing that this can do is not to kill the virus, but it is going to damage our normal cells. That's what happened with the SARS disease. So it's damaging our normal cells. This is what I'm going to show you again. Here, if we see, this is the normal alveoli, and it has this epithelium, and it has these alveolar cells, and it has this surfactant, that is a small mucus layer which is reducing the surface tension. Then alveoli can expand easily. It's not tight. It's surface tension. It's slow with it is called surfactant. So it is normal. When the virus have done all those works, and then it's reducing its the cell's response against that is innate immunity also. And when it is released, when our innate immune system release cytokines, interleukins, interferons to toxic to the virus. It is toxic to the virus. But these inflammatory mediators, innate immunity mediators, now going to damage the cells, normal cells. It's going to damage the normal cells. Virus escape already. So this is what is happening. And here the more important thing, these have ACE2 receptors. ACE2 receptors has a very good function, but when virus attach to that ACE2 enzyme and ACE2 receptors cannot function normally. When ACE2 is not there, angiotensin 2 is not going to bind to ACE2 and produce that. I am going to tell you something. I don't have a diagram here, but when angiotensin 2 has two options, it can either bind to ACE1 receptor or ACE2 receptor, but mainly angiotensin 2 enzyme bind to ACE1, not to ACE2. To a small extent is bind to ACE2. When it is bind to ACE1 receptor, it's going to produce blood vessel constriction. Blood vessel constriction in the microvessels or adjacent to this alveoli. And more than that, it's going to circulate in the body also to affect on other blood vessels. And also it's lead to some fluid leak also. Fluid leak. And then lung edema is also there. So when ACE1 action is more, we get more of the fluid leak. When it is bind to angiotensin 2 bind to ACE2 receptor, where this virus is also binding and blocking the function, ACE2 receptor is going to get the different chemicals are producing. It's going to really bradykinins and then angiotensin 1 to 7, and those chemicals reverse the action, has the reverse action of angiotensin 2, the vasoconstriction. It says vasodilatation. Vessels dilate. So that action is not there. So the blood vessel constriction action, fluid leak action is going to reverse by angiotensin 2 receptor function, and that's cancelled. That's cancelled by the virus. And then more than the receptor binding, the virus can damage our ACE2 enzyme also. So then that function is not there. That's why people are getting lung edema, that is lung swelling. That is because fluid leak from angiotensin 2. And there's fluid leak. In addition, when the inflammatory chemicals are there, it's going to produce inflammation also. So inflammatory secretions are also there. They increase the blood vessels permeability to remove more and more fluid from the blood vessels into the tissues. So by that way it's going to give swelling around the alveoli. And other one is that this ACE2 is mainly found in alveolar cell type 2. Alveolar cell type 2 is the one that is producing this small blue color one, surfactants I said. So this surfactants function is to easy to expand. When this white color area is the one that we get the air from the bronchus, from the outside, it's filled there. When it is filled, this blue color is the one that facilitate there won't be resistance. It's easier for this alveolar to expand. That is like a sac that is expand easily. So when the virus attack to the type 2, that's going to get ill then. Type 2, that is the one produced in this surfactant, this blue color mucus like secretion. That's not going to produce. So then the air cannot get easily expand this alveoli, this sac. So we feel it is difficult to breathe. Then it is difficult. We have to get an effort to expand this. It's very difficult. So it is tough. It's tough, like it's not easy as earlier. So when the virus attack, that's why we get shortness of breath. That's what we call. That's why we ask people, do you have fever? Do you have cough? Do you have difficulty in breathing? The difficulty in breathing part is here. And also cough is also due to these chemicals released from the virus inflammatory mediators. And the fever is also due to these chemicals. Once it enters to the brain hypothalamus, it's going to sense it and we get fever. Our thermostat is changing. So it's going to a different level of and we get fever. Thermostat change. So that is some knowledge about it. And so this is what is happening. And then there's some basic understandings there for virus. Now I'm going to tell something about this coronavirus infection, and then I am going to some epidemiology here. And when we are testing, people are talking now about antibody test and antigen test. That really we are doing PCR test. PCR is polymerase chain reverse some test. PCR test. So PCR test is going to detect the virus, whether it is live or dead. It can detect the PCR. What is it going to do is the mRNA of the virus genome change to DNA at the test, and then that DNA is going to recognize by the test. So that is called PCR test. With the PCR test only, we can find whether virus is there. Virus is there. That is this maroon color one. And serological test is when there's immunity. I said that the different flag is there. So then body is going to develop antibodies against this virus attack. So these antibodies are shown here. They are the one detected by the serological test. But serological test won't tell whether there is a recent infection or whether it was a past infection. When both are there, only we know there is a recent infection plus immunity development in the body. So this is what I am trying to show here. And this is some epidemiology, how the virus has gone. So this is basically this one. So the bat is the reservoir of this virus. Bat has lot of coronaviruses, about hundreds, 500s viruses, but it cannot directly enter to a human. It's not easy. It's not easy. So the reason is this bat virus spikes its key is fit into a different lock here. It's like a triangle. For you to understand, this is like a triangle. Say it is triangle like lock. So, but in the human, it's a circular like lock. So this spike, star-like spike, has to change to a circle, sphere, half a hemisphere like appearance to open up the human cell lock. This key has to change to human cell. So the scientists, virologists think this change, this one to change to this hemisphere like appearance is not really hemisphere. That's why I'm going to, this is to show you some different configuration. To this configurations happen in another animal. So by this may be, they are telling some animals are there that the Chinese people used to eat, and some wild animals in that they are thinking that in that farm there are various, and the Chinese people think it's a medicinal, something like some small animals which very rare, people don't eat, but they think it's medicinal, but it contains virus unfortunately. And that one may have changed the virus into this level, which is good enough to open up our human cell. So that is the intermediate host has entered to humans in like that. Once it is entered to humans, then the virus genome change enough to human transfer. So this is what is telling here. Okay, now more on to epidemiology, public health science. So when we call this, we see that it's spreading fast. So when we know about one person, about 3-4 days later, they tell now three people are infected. So then another 3 days later they say no, 9 people, more than that, 18. It's like that. It's spreading like exponential way. That is calculated, called R0. This is R0. R0 is the one for COVID-19. It is 2.5 at the moment. In the USA, when it is spreading in USA, it is at 2.5 number, R0 number. So that is to say, from one person how many other people can get infected with COVID-19 is 2.5. So that's why it is going fast. If I can see from 1 to 1, then at least it's going to flatten. There won't be much of, we won't hear is multiplying. And if it is, we are waiting to hear it is going less than 1. So then that is the time that this virus, a pandemic, is going to go down. So this is F0. You can, so we have to think of what is this R0. Sorry, R0 is depending on infectivity and infectious period. Infectivity is that how long how many people can spread, and infectivity how fast, how efficient the spread of the virus. So it's depending on the number of contacts by this person, say this is the infected person at the beginning, then how contagious the infected person is. Contagious. So now it is, when it is SARS-CoV, now it is called SARS-CoV-1 because now COVID-19 is SARS-CoV-2 virus. The name is SARS-CoV-2. So the SARS-CoV has changed to now SARS-CoV-1. Now SARS-CoV-1, that is 2002, when the epidemic is going on, as SARS epidemic, then the distance of the one person's aerosol can go to other person was just 3 feet. Now it is 6 feet. They found 6 feet distance. It can go to 6 feet distance, and that the 6 feet distance person get adequate viral dose to get infected. Adequate viral dose get into at the 6 feet distance. So that's showing the how virulent the virus and how lower dose of virus is enough to infect the other person 6 feet distance. So it is contagious. So another one is that I have to tell you, still not that much, but the ACE2 receptor is abundant in very much more in both the lung alveoli and also lung tract, respiratory tract, and also in small intestine. Small intestine is small bowels. So it can go through the bowels also. It's the fecal route is also possible. That is also other contagious. It is contagious. And the touch surfaces, that how long this virus can stay on a surface. It's found even cardboard, it's 1 day, and on a glass or some stainless steel, or 2-3 days also. So it can stay there till that time. Still it is capable of entering into a human. That is what we call survival time of that virus. So it is capable of entering to a person at that time. So that's what we call, when we touch that and touch our nose, it's going to enter into our respiratory tract. So that is contagiousness. And the number of contacts. So how this person is like somebody who is very in a church father or somebody, then he can give it to more. Somebody who's giving lectures, and if he is infected, he can give it to low number of people. So depending on the persons, or if he is going to places, the public places too much, then the infectivity is high. Then infectious period is the other one. Infectious period is the how long the patient is disseminating this virus. So when he die, he don't. So once the infectious and unto death, or once he get the infection up to the recovery. And there is one more thing. The infectious period starting the SARS-CoV, that is during 2002 SARS epidemic, they could have infect after getting symptoms also. 24 hours to 36 hours of getting symptoms. After that only it could have infect to a other person. But COVID-19 is totally different. During the infection period, last 2 days he can infect to other person, and infect to other person. And also there are some people, they don't get symptoms. SARS-CoV, symptomatic people could have only give disease to others, but there are some people asymptomatic. By the Japanese study found, and also the Diamond Princess ship studies found this 51%, around 50-51% people are asymptomatic, yet still they can spread the disease to other people. It can spread to other people asymptomatic. So this is a challenge with COVID-19. So this is what R0 is higher here, higher, and So this is what we can think of, how can we do with R, how we can reduce the R. That's what our purpose. So the best is to vaccines. With the vaccine, this infectivity is going down. And the other one is a drug. So we need not to stay the death of a person or natural recovery. We can accelerate the recovery time with the drug. And with the drug, we can reduce the virus load, and we can reduce the. There are some experiments with hydrochloroquine in France, and they found reduction in virus load after treating this hydroxychloroquine. There are some viruses, antiviral drugs also. They still are testing. It's not confirmed yet. Recovery. If we reduce the recovery, then we can reduce the R here. And some people are thinking of herd immunity, but the herd immunity in COVID-19 is dangerous to think of that. It's natural herd immunity, because then that means the virus is uncontrolled level of spreading and a lot of deaths, because there is at the moment in America it's 3.5 mortality rate, and that means the millions and millions people are going to die once we get the herd immunity. So that's what I want to tell you about this R0. That's why epidemiologists are trying to do something, but we can do physical distancing. That's what I showed you earlier. And the contagious people should not have ceremonies and functions or lectures or sermons like that, or musical shows, parties. And the contacts, then we can reduce by that way contacts. And also we have to find asymptomatic people, those who are spreading the disease, and improving the recovery rate of the people, with isolate them and follow them up with their status of the virus and like that. So this is some basic epidemiology and basics about the virus for you to understand about the COVID-19 differences from SARS. So thank you very much for listening.